Okay, so I guess you can hear my voice. If you can hear me, please shout on the chat. Then I can see you. This is all working. I think at least one person always shout on whether you can hear me or not. Okay, so that's great. Great response. Okay, thanks for your participant. Because sometimes the Zoom ask me the installation just before the class, so I'm not sure whether the installation is working or not. But anyway, okay, so back to the class. And before we move on to the today's session, bit of announcement, the first exam. So we have 2 more sessions to go before the first exam, and that will happen on this week. And we're going to have the quick review session and Q&A session. And that's going to be the next Monday, and your first exam will be on the next Wednesday. So that is sort of the brief schedule this week and next week. And I guess the topics from 1 to 4 is pretty much the vanilla. So I mean, that's why I thought not many questions from you on Slack, but the topics about the gene and heritability is bit something could be tricky. So if you have any questions, it's always welcome to leave on Slack. I mean, I want to emphasize at least before the Q&A session that then you cannot be late on the exam preparation next Wednesday. So that would be great. Okay, so back to the today's topic. So we're going to learn about the gene and how this gene is perceived at the first time in early genetics, and how we can link this idea to the Mendelian genetics and the late sessions on the molecular biology or the population and mutation topic. So we're going to learn about the 4 things today. So first one is the definition of genes and the relationship between genotype and phenotype. That is sort of the first part of the lecture. And also we're going to discuss about the components in a gene. And I think this is pretty much overlapped with the molecular biology topics. And we're going to discuss how Thomas Morgan's experiments moved the perspective from chromosomes to a gene concept. And that is the last topic we're going to deal in this session. And the same disclaimer and notes. Okay, so the first topic. So the first thing we need to discuss is how the concept of gene is only defined in early times. So as we discussed in previous session, gene is a core unit of the heredity, and that controls the development of the trait. And this sentence is obviously mixed between the gene and allele. If you want to go detail about the Mendelian factors, but the situations when we discuss the heritable factors between the early 20s and the molecular biology era around the 50s and 70s, the gene is actually a core unit of the heredity, because the people thought about the relationship between the genotypes and phenotypes in the center of the gene flow. But as I keep mentioning over the many sessions, the meaning of genes is actually differed by time. And as we know more about the biology, then the concept of the gene is slightly differently perceived over the many different contexts. And one thing we can obviously say that the Mendel did not know about the gene, and his idea about discrete particle in his model, in his particulate inheritance, is actually indicating the concept of gene or allele, right? So this is sort of the early genetics we can talk about in previous session. But anyway, in early, Mendelian people, they, came up with the idea about the concept of the gene. And Wilhelm Johansen was the first person who introduced the term gene, and he tried to describe how Mendelian inheritance working in the, in biology and inheritance, and how these Mendelian factors is working in the notion. But obviously, Johansen didn't know anything about the molecular biology, which is the, the translation between the DNA sequence to protein sequence. So, I mean, he didn't know the details of the gene. I mean, so the early indication by Johansen was actually the meant to be a allele rather than gene. So you probably have been, perplexed concept about the gene and allele when you read the, text in the, describing the early 20s and I mean, probably the mid 20s as well, because there is a sort of mixed use of the gene and allele at the time when they tried to explain the Mendelian inheritance. But in more modern sense, especially in a large scale genomic region, the genomic research in these days, we call the Mendelian factors allele, and this allele is a genetic variant and DNA sequence changes, then we can link to the phenotype. Okay, so Johansen also create the terms called phenotype and genotypes because he has to explain the relationship between the Mendelian factors to the phenotypes Mendel described at the time, right? So the phenotype is a sort of the entity of we see sort of external appearance from the organisms, right? So whatever the peas are tall or short, wrinkled or round, and all of these, the externally we can perceive from the organisms we call the phenotype, right? And also again, the phenotype is a sort of a different conception over the time. So, I mean, nowadays people thought about the phenotypes like the biometric things you measure from your Apple Watch or whatever from your iPhone. That could be all the phenotypes we can count in many different biomedical experiments. But anyway, at the time, the phenotype is normally describing the situation we perceive from the external appearance, right? Okay, and also Johansen introduced the term gene in his book, so it's a bit later than the phenotype and genotype. And this is the this is the term originally supposed to describe the situation in an opposition to Darwin's theory of pangenesis. So Darwin's idea was on the mainstream at the time. So obviously the Mendelian people, they are on the opposite side, and they try to argue that the Mendelian factors is correct in inheritance situation, and it has to be appreciated rather than the Darwin's propose about the pangene and pangenesis. So that is one notion why the Johansen's the terminology at the time was called gene, right? And then in 1905, William Bateson coined the term genetics from the Greek word called geno, right? And that means to give birth, right? So it's literally meaning the inheritance phenomena. And he tried to describe the all disciplines or studies to see how the parents and child resemble to each other, and how the variation can happen in a population, all about the inheritance things. He wants to call it genetics, and that area of the study we call it genetics, even nowadays as well. Okay, so there was the bit of the short history about how the definitions of genes and related terms evolved over the time. Okay, so we also have to think about more modern context about the terminology genes, because the perception of the term gene is changed over the time. So the first time was the Mendelian factors and the Mendelian inheritance re-evaluated by Mendelian people in early 20th. Then people, move into the central dogma, the concept mid 20th. And at the time, the Mendelian factors is slightly differently, perceived by the molecular people. So they start to think about that idea about, hereditary factors proposed by Mendel is actually the units can produce the RNAs or proteins, and that proteins or whatever RNAs can work on the phenotypes, right? I mean, it's more onto the proteins, but anyhow, the people thought about the sort of the workflow from the DNA to RNA to protein, and how this, the gene is working in an inheritance situation, but also in the protein expression as well. So central dogma was at the time. So if you think about the today's topic about, the concept of gene, you can actually appreciate that the different, transitions of the perception of the gene over the time. So from the, the hypothetical terminology at the Mendel's time, then people see chromosomes as a Mendel's idea and gene as a Mendel's idea, then to the whatever the DNA sequence as Mendel's idea, right? So as I explained that the after mid 20s, there are some, the notions about, other than proteins, people also thought about, the RNAs, the intermediate units RNAs can work as a the final functional unit. So I mean, another areas arising from that thought. So we thought about the we thought about the non-coding RNA at the time. So these two things are actually formed around the central dogma idea. And then after that, the people developed the latest technology called genomic sequencing, and people see the sequence on our genome, and people find the changes which are equivalent to allele at this time, and people link that alleles to the Mendelian factors. So that is more recent definition of a gene defined by Mendel in all Wilhelm Johansen in early times, and the Mendelian factors in Mendel's days. Okay, so we have then from this idea we can have more questions about the human genome and the concept of the gene. So first question we can ask that how many genes in human genome, right? So I mean that could be quite natural, naturally occurring curiosity when you study biology, right? So I mean, many of your friends, or not many, maybe few or some of your friends or family members ask you, oh, you study biology, so do you know how many genes in our genome, right? So nowadays we know the answer, right? So we can search the internet, or we can go to the link as I put it in here. So this is the database of the description of genes in our genome. So if you go to this link, the database called GenCode, and on the version 37, we can actually see the statistics of the genes we currently knowing from our genome, right? So the total number of genes in human genome is about 60,000, right? And out of these 60,000, the number of genes that can actually produce the proteins, this is around 19,000, right? And the rest of them, which are about the 40,000 genes, is whatever the non-coding RNA genes or pseudogenes, or the genes related to the immune, the defense system, which are working in the COVID-19 or other sorts of the influenza systems, right? So now we know the statistics on that, and also looking at other the information about the transcript and or the whatever the translation situation too. Okay, so if you are interested in that, you're going to this link. Okay, and nowadays genes are perceived as a unit that can make a certain protein and determine a certain trait, right? This is the sort of the basic notion of the central dogma we learned from molecular biology. And as we explained in previous website, in human, there are about 20,000 genes that can make proteins, and most of them are involving in the determining trait, right? So you can actually developing that questions like how many genes determine height, or how many genes determine certain disease or some blah blah blah, and that could be a sort of starting point. You can think about the genes and disease or the trait of your interest, right? Okay, and also one interesting part is that the many different types and functions from long evolutionary process. So genes are actually the place having the implication about how human beings or many biological organisms has been embraced or faced the evolutionary process from the long history, right? And the genes has that sort of the signature from those process, and that is also quite interesting things. And also, if you go into this database, you might find why so many database in the field, because everyone wants to be ahead. So people make up the different database, and they looking different sorts of the aspects about the same topic in this context, the gene. But anyway, so back to the question is how many sort of the functional groups of the gene in our genome, right? Because we have 20,000 genes, but probably tens of genes or hundreds of genes are working in brain, working in brain cells, or working in kidney, working in your heart, working in your nails, or blah blah blah. And in that case, you can go to this database called HGNC, and you can search that gene group because here is a list of the gene group index. Then you can search from here. So whatever you want to search, the gene group, my favorite is the sodium channel. Then you can search the sodium channel, then see which genes are actually belonging to this portion, right? So click the sodium channel, then you can see that how the genes have the functional relationship between the biological terms and how many genes are actually within these functional groups, right? So all of the information you can easily search from this search of the database. Okay, and one interesting part is that some genes can be functional, or known to be functional, but other genes are still unknown, right? So that's why we're still doing research, right? So I mean, from human genome, we know that there are 20,000 protein coding genes, but we are actually know the functions of the genes, most of them, but still few are missing in terms of their functionality in biological system. And also we didn't know many things about the non-protein coding genes, so which are about like 40,000 genes, and why we have so many 40,000 the numbers in our human genome, and what that means in the biological context. So in the sense, we still don't know many things about the the functions of the many genes, and the research is heavily going on to that direction. Okay. And then also one thing for the Korean students is that the alleles and the genes are not identical concept, right? So as I explained many times, the many Korean translation lead incorrect information as to allele and gene, because they are literally quite similar words, right? So these two. So because of this the stupid the translation, the many of whatever the media contents or even like, you know, professional stuffs, they are missing the information and brought the missing information to the public, and it's always been a, you know, an issue in biology topics. So please keep in mind that these two things are quite different, could be quite different meanings in different contexts. So has to be cautious for the proper use. Okay, so back to the idea about the central dogma in molecular biology, and I'm pretty sure most of the second year students, they are working in the molecular biology course they're taking, and you're studying this concept over the semester, right? And you're going to learn about many details and mechanistic differences between the process and stage and how they are, or which units are working in each stage, right? Basically, the idea about the central dogma is the workflow from the gene to protein, right? So gene to protein. So what that means in the genetics or the broad sense of the biology. Okay, so the process from the gene to protein is actually corresponding to the genotypes to phenotype, right? So when you see some phenotype, when you see some organisms, certain traits, certain external appearance, we are defining as a phenotype. So that phenotype is constituted by proteins, right? So that proteins is actually modulated by the RNA or the gene, and that is the basic concept of central dogma, right? So when you, so I mean, some situation, I asked the questions to the students like, you know, what is the meaning of the central dogma, and or the questions like, you know, what is the central dogma, and people often, you know, technically describe that answer like, you know, central dogma is the process describing the DNA from DNA to protein. But I think this is not 100% correct, because the why we are caring about the central dogma is that we want to see why organisms have that phenotype in their, you know, whatever the external appearance or the behavior process or whatever the physical process, right? All of these, the biological phenomena we can describe from the organisms is actually explained by the molecular mechanisms, right? And that formula we call the central dogma, right? So whatever the situation, like, you know, why you are tall, why your height is about 190, because some of your proteins is lead you to have the higher, you know, height, right? Higher stature, right? Or some situation like, you know, why you are prone to have more alcohol consumption. The reason is that some of your proteins actually leads to make you to do that behavior, right? Because like, you know, the whatever the neurotransmitter or the proteins in your nerve system actually led you to do that action, right? So moving that perspective from the whatever the situation, you know, we can abstractly describing that activities. We nowadays we actually see the backbones of that actions or behaviors with the concept of the mechanism, the molecular mechanistic way, right? So one thing, I mean, could be quite, you know, broad or the rough topics onto that, but you know, some of the situation like, you know, the disease like schizophrenia or bipolar or depression. We might ask in this century, like, you know, why the person has that phenotype, right? So some people thought that, probably in 2 centuries ago, or like, the around 50th century or 10th century, so a while ago, or the time of the when the Bible is written, people thought that that is the sort of the possessions of evils, or that person is wrong, or something, because there was the sort of subjective way people thought about, or judge about the things, right? But because of the scientific advance and more knowledge on to the, the biological activities, now we can describe that the schizophrenia is the issue of the neurotransmitter in nerve system, and the neurotransmitter, it has a genetic changes in a DNA sequence, and they can lead to the abnormal the protein expression or the protein homeostasis, and that can ultimately modulate the typical mental health activities in our brain, right? And we can approach more mechanistic way than thought about the situation rather than a person, right? So I think this is great transition from the mundane society to more modern era, because we can then we can start thinking about how we develop the treatment, or how we develop the better interventions, or how we develop the better approach to dealing with these peoples, right? Rather than just abandon them people from our society, or whatever, you are the useless people in the society. We can actually change our notions to the help those peoples, and also thought about the advanced way to the dealing these topics, right? So in many disciplines of the biomedical sciences, we're actually looking at the mechanistic way how diseases are arising from the whatever the genetic underpinnings or protein underpinnings, right? And then we can thought about the more reduced way to develop the treatment and the intervention way to improve their life, or improve their quality of the mental activities, or whatever the health conditions, right? So you always keep in mind that your study about the central dogma is always linked between the genotype to phenotype, and those phenotypes is the one we could face in everyday life, and also these are all part of that source. So that is one implication we keep in mind. That okay, so genotype I had a long whatever blah blah blah, but the genotype is called the part of the genetic makeup of a cell, and therefore an individual which determine one of its characteristic, right? The characteristic is the meaning equal to phenotype, and phenotype is the set of observable characteristic of an individual and resulting from interactions of its genotype with the environment, right? So the easy, simple examples about the genotype and phenotype was the Mendelian notation, the dominance and recessive, and the phenotype is the P color, right? Or the other way, I could give you an examples of the SN2A gene, which is the sodium channel alpha 2 unit gene, one of my favorite. And that is the gene located somewhere in our chromosomes and translated into the protein called the sodium voltage K 1.2, right? So this is the protein name, or we can simply put in this one as the protein name. Okay, so the genotype we can thought about is that the SN2A has the long stretch of the DNA sequence, but one place I want to focus on, which is the position of the 1013th DNA position, and that has the DNA sequence has G and G, right, on both strands, right? That case, that individual have the phenotype, which are the brain development, right? So how this SN2A is working in our brain is that the SN2A is the sodium channel which transfer sodium ions from the external cell layers to the cytoplasm. So it modulates those ions beyond the cell membrane, and it actually working on the brain action potential, which is the signal systems of your the thought, cognitions, emotion, or blah, 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 all of the things happening in your brain. And when the sodium channel is working properly, those ion transmissions working well, and all of the brain functions work as expected, right? Okay, so the structure of the SN2A gene is like this, the membrane protein. So membrane protein means that it's sitting on the cell membrane, right? So this is the inner cell area and this is the outer cell area. So that's why we call the extracellular, and this is the cytoplasmic. And the SN2A is embedded in this cell membrane, right? So lots of segments and the link between these segments, right? And this segment is wrap around and forming these types of the pore, right? So this is like a tiny pore. So there is a hole in the center of the protein, and the sodium channel, which is the Na, is going to this area, right? So this area is equal to this hole. So your sodium channels outside your brain cells is going into this process, this direction, right? And some of the link between these segments has the filter, and that actually working as a whether you want to take more sodium channels or not, right? So working that way. Okay, so this SN2A is important in your brain development, right? So when you have the wild type, which is the most of the population has that sequence, right? And that is the bring you the normal development of your brain. But when you have the T sequence in your DNA, which we call the T allele on your DNA sequence, that case you have the stop codon, which is that your SN2A protein translation stop at this position, right? So SN2A has the long DNA sequence is about like 40,000 base pair, but once you have the T allele at this position, then you're going to have the stop codon, and your protein sequence is truncated at this position. One thing I should correct my explanation, which is these numbers. This number is not the DNA position. This is the protein position. So SN2A protein has about 2,000 protein sequence, which is the 2,000 amino acid, right? So this position is about the middle of that SN2A protein sequence, right? So when you have the T allele at some position, then your amino acid, which is the glycine, is changed to the stop codon, right? So this G is not DNA nucleotide G. This is the glycine, right? So that is changed to the stop codon. So no more amino acids translated after this sequence, right? In that case, because of this one DNA change, your SN2A is corrupt, and your SN2A is not working properly. And because of this malfunction of the SN2A, that individual are going to have developmental disorders, because SN2A is critical in early brain development. So that can lead the normal development. But once you have some stop codon in that protein, then it's going to have the developmental disorder, right? So one thing you might ask by yourself is that how we can represent the Mendelian notation in this situation, right? So you can simply, or you can present the abstract way, like large A or small a, large A and large A. Okay, so for the normal development is the large A and large A, right? Or here is the large A and small a, because T allele is something different from the the most of the population, right? So that case you can expect that there must be different notation. Then the second question is that which allele act as a dominant, right? So that is the another question, right? So we are not dealing with the dominance is the frequency of that allele in a population. Dominance is nothing to do with that. We learned it from the Hardy-Weinberg principles, right? So the concept of the dominance, it has to be understood within the context of the molecular phenotype, right? So think about the situation. When you have that the T allele in this position, then you're going to have the developmental disorder, right? So this is the obvious phenotype. So we could say that this T allele is dominant to G allele, right? So T allele should be dominant, right? The reason is that this individual has heterozygous genotype. So one is G and one is T, right? So T allele is expressed, and this T allele is totally masking the G allele, right? So T allele is the critical in terms of measuring or judging the phenotype. So this case, T allele should be dominant to G allele, right? So then we can say that we can judge from this heterozygous situation that the T allele is leading the dominance in the phenotype, which is the normal brain development, right? And G allele is recessive to T allele, right? And then again, as I explained, that the G or T, its proportion is nothing to do with the proportion of these alleles in a population, right? So because the frequency of this G allele is way higher in a normal population, in a human population, and this the number of individuals having this T allele is way smaller than the G, right? So the frequency of the G or T allele is nothing to do with the concept of the dominance. It's only the meta when we thought about the molecular perspective, right? So if you're not clear about this explanation, please leave your questions onto the Zoom channel. Okay, so what types? So the questions from the Saren. So what types of the developmental disorder does the GT genotype result in? That is interesting question, and not really the matter in the context, but if you have this mutation in your genome, then it leads to the range of neurodevelopment. So I mean, it could be the epilepsy, it could be autism, it could be intellectual disability, but the reason is that SN2A is quite pleiotropic, so that means that diverse function in development. So we couldn't really pinpoint that one disorder is occurred by this mutation. It actually impact on or result in many different development. But anyhow, the stop codon is working in that the phenotypic development, right? Okay, I mean, I hope it could be answered for your question. Okay, so I mean, if you're not clear about this sentence or the explanation on this part, I mean, feel free to leave your question onto the Zoom channel or the Slack. That would be welcome. In the meantime, I continue to discuss the later section. Okay, so let's zoom out the chromosome 2. The reason is that the SN2A is located on the chromosome 2. So we have the karyotype image like in here, and we can select the chromosome 2, metrocentric and mesocentric, and zoom out to see the idiogram. Obviously, if you zoom out this, zoom in this, you're not going to have the idiogram, but I mean, it's equal to idiogram. So if you look at this idiogram, and if you go into the 2q24.3, then you will find the positions where SN2A locates, right? So SN2A is located on the chromosome 2 and around 166 million base pair positions, right? And SN2A is sitting on in here, right? And if you want to see SN2A on a genome browser, then you can click this link and you can travel to chromosome 2. And it's quite slow. The reason is that the UCSC brought us to the US server. Okay, so going into here, then you can see the same ideogram and see the SN2A on here, right? So you see the gene's position, then you can obviously zoom out the position like in here. So you can drag the position and you can click zoom in, then you will see the positions of SN2A like in here, right? And we actually see more things from this image. So if you zoom in there, then I mean, literally you see much details of the SN2A like in here, right? And you see the positions of that. And you can zoom in more, then you will see the compartment sitting in the SN2A. This compartment is, I mean, the same notation across other, you know, the human genes or whatever the biological organisms genes. So, I mean, no matter which species coming, we always use the same notation, which is like exons or introns or promoters or something. So all of these compartments, all the organisms in our planet, we have the same things, right? So we can also see these things in our browsers as well. So you see the SN2A gene here, and SN2A gene here, SN2A gene here, SN2A gene here. Then these genes has the exons here, and introns here, and exons here, another exons, because the genes can have multiple exons in a gene. And also, obviously, if you have multiple exons, then you have the introns, right? So for this, we have to know what is the exons and introns, right? Okay, so the first thing is gene contains exons, introns, UTRs, promoters in its transcript, right? So you have many concepts about the genes in this sentence. So the first thing we have to think about is the gene can have various transcripts due to the alternative splicing. So when you see the genome browser for SN2A here, you might find interesting because the SN2A has multiple appearance in this browser, right? The reason is that the SN2A is expressing multiple different transcripts, right? So the transcript is the units of the different combination of exons during the gene transcription. The reason is that the having multiple transcript can actually work in a different molecular situation. So for instance, SN2A is expressed in the brain, but also SN2A is expressed in your heart, right? Because the SN2As work as a, the action potential or synaptic, signal transmission in your brain, but also it delivers the signal transmission in your heart, because the brain and heart has the signal systems in their organs, right? So different transcripts work in a different situation, and also different transcript work in a different developmental context too, right? So when you're young, you have different forms of SN2A, or when you're adult, you have the different forms of the SN2A, right? The reason is that the humans or most of the biological organisms has to adapt in many different environmental contexts, right? And also doing the many different activities, right? You do many, the high level cognitions or high level emotions, right? And that situation, you have to use many different cards of your proteins. That means that, you know, if you have only 5 proteins in your body, then you cannot do many different, you know, activities, because your activities is, you know, fell into 5 different categories, right? But since you have 20,000 genes, and also you have the, you know, large number of transcript and different composition of this large number of genes and transcript, you can do more sophisticated and more complex activities, you know, chimps or mouse cannot do, right? So think about the situation. And also you probably learned about the why the evolutionarily higher organisms are having the alternative splicing system, because the evolutionary advantage over the history, right? So that's why when you look at the, you know, genes, you always have to, you know, to link your idea to the various transcript. Okay, so anyhow, each transcript has the different compartment, which is the exon, introns, and also UTR, right? And also promoter. So exon is a region of transcribed gene present in the final functional RNA molecule. Wow, long sentence. And intron is any nucleotide sequence within a gene that is removed by RNA splicing during maturation of final RNA product. Or you can simply think that the exon is the area of encoding or producing the protein amino acid sequence, right? And the intron is the area, genomic area between these exons, right? So between these exons, there is a long stretched genomic region, we call it introns, right? And UTRs, this is the untranslated region, and we have the 2 sections, which are 5' terminal and 3' terminal, and that's why we call the 5' UTR and 3' UTR. And you probably learned in molecular biology, and this 5' UTR is close to the transcription start site, which is the starting point of the gene transcription by RNA polymerase II. And that region is also containing the upstream sequence called promoter, right? So this is the area promoting the gene transcription, right? So all of these area, or we call the whatever the transcription start site units, that is the constituted by the promoter and the 5' UTR. And that is the close to the first exon, right? And the 3' UTR is the last one, and also close to the last exons, right? So the questions about the transcript is that the first one is the transcription is always acting as a 5' to 3'. Yes, it is. That's how biology is working. And if you're not from this planet, probably. But the other way we have to think about is that the DNA has the double strand, right? So when you present your gene, you can present like this way, or you can present the reverse way, right? So the first thing is when you when your gene on the forward strand, which is the first strand, that is the 5' to 3' direction present in this way. So this is the arrow indicating the direction, right? But if you go to other genes, let's find out which gene is the reverse direction. Some of the genes have the reverse direction. Probably this one. I have very eagle eye. Come on. Okay, let's drag it. I practiced this moment over entire my life. Okay, very sharp dragging. Okay, FIGN gene has the reverse direction, right? So that case, this gene sitting on the reverse strand, and that has the 5' to 3' and like in this way, right? I mean, because of the the genome browser only showing you the haplotype, so haploid genome. then it actually has the both strands on this visualization. So the only way we can represent that directionality is this arrow, right? So keep in mind that this gene has the reverse reverse direction of the gene transcript. Okay, cool. So fun genome browser. Okay. So I think we cover everything on here. Okay. And also the number of exons is quite different by genes. So some, gene has many exons, like 100 exons or 50, exons, something. But some genes are quite simple. So some genes like, only one exon. And there must be a reason for that format. I mean, in the context of the evolution or the biological functions. And if you're interested in that, search the situation like a single exon gene and why that gene has to have single exon over the long evolutionary history. There must be a drama onto that situation, so you might find fun. Okay, so last chapter of the today's lecture is the Morgan experiment, and this is actually well covered by high school biology. So I'm not pointing out the same topics for that, but we can recap what Morgans did between the Mendelian genetics to the molecular biology. He's the one of the key figure we have to think about in that area, right? So the first thing is the well-known things about the Thomas Morgan was he used the fly experiment looking at the Mendelian factors on chromosomal behaviors, right? So that was one thing. And one interesting part is that the Morgan actually rejected Mendel's idea because he had been trained in the context of the blending inheritance. And he was definitely the side of the blending inheritance at the time. And he was the person who strongly believed that gradualism in the evolution. So he definitely sort of the person from the biometric, or I mean, he's not really biometric because he's not a mathematician, but he's definitely, into the, he's definitely a person into the blending inheritance patterns because of his, the training background, right? Then but Thomas Morgan was a person who strongly believed that the natural experiments, right? So he wants to believe what he did experiments, right? So he tried to explain that blending inheritance using the fly experiments, and he wants to, manipulate that situation by his hands, right? So that's why that was one of the motivation he was falling into that direction. Okay, so what he found, so two things. So first one is he conceptualized the gene on the chromosomes, right? So he's the person moving the perspective from chromosome to gene as to the Mendelian inheritance, right? So he was the person contribute that notion. And the second is the genes linkage, right? Genetic linkage on the same chromosomes, right? So his idea about the linkage was the great exception of the Mendelian assortment idea. So and that is also the link between the Mendelian factors to molecular biology, right? And also Thomas Morgan's finding was, giving great inspiration to other Mendelian people, and they are quite excited by this fly experiment, and they followed into that direction too. Okay, so how Morgan chose the fruit fly for his experiments? You probably learned from the molecular biology or other, the general biology course. We use the model system to study biology because, especially in an early 20s or mid 20s, we have sort of short of the technology to see or to describe the biological phenomena at the time, right? So we have to use quite the efficient system to produce the lots of generation that can you see quantitative way to measure the phenotype, genotype, and also use the system we know about their genetic backgrounds, right? So I mean, they know about the chromosomal background, and also they know about the the fly has the 4 chromosomes, and you can easily infer something, the genotype information to the phenotype information, right? And also it's based on the diploid, so it's and diploid, and also the distant systems on the sex chromosome, so you can easily follow up the sex chromosomes genotype to the phenotype. So all of these characteristics is giving an advantage to fly experiment in Morgan's idea, and that was quite working well at the time, and also in these days as well. So using a model system is always giving you a new dimension to describe the biological phenomena in our field. Just TMI, but recently I'm working on a prayable, which is the one of the cutest animal in the world, and it's the monogamic behavior in a social system, and we feeding them oxytocin and trying to explain how the autism behaviors is working in that cutest animal, because they are socially well constructed behavior, and we couldn't get that advantage from other model animals like rats or flies. So always digging into the new types of the model animals for the biological research is still valuable and still doable, and still lots of people wants to do that in the future generation. So if you are if you feel that I'm a druid of the animals, then this is your field. So go and search and think about which animals is sort of the hipster things in this generation, and which topics are totally new to the field, and that is always the exciting questions. But anyway, back to the Morgan. So Morgan noted the wild type or normal phenotypes that were common in the fly population, and that is sort of the basic assumption that the wild type and the most of the individuals have the constituted in the population. Okay, and then the traits alternative to the wild type, he called it mutant phenotype, right? So this is distinct and opposite situation to the wild type, right? Okay, so what Morgan found that he got the mutant, he defined the mutant as an organism or a new genetic character arising or resulting from an instance of mutation, right? So in the other days, in one experiment, Morgan mated male flies with white eyes, which is mutant, with the female flies with the red eyes, wild type. And F1 generation all had red eyes, and he concluded that red eyes is dominant according to Mendelian factors. And the F2 generation showed 3:1 ratio red-white eye, but only males had white eyes. So then he thought about that probably the white eye mutant alleles must be located on the X chromosomes, right? And this is the sort of the supporting idea of the chromosome theory of the inheritance, and bring the chromosome idea into the gene level, right? So there was a sort of background of his experiments. So as I mentioned, that he used the red eye flies and white eye flies, and red eye flies is the mutant, right? He didn't get this mutant in initial stage. He accidentally get this red eye mutant from his experiment. So when he did experiments in using flies, he has to do in a quite isolated and dark room, and he generates so many flies over many years, right? I think it's about 3 or 4 years, and one day suddenly he has the red eye flies from his fly population, right? There was shocking things, because he never thought that the flies could have the red eyes, right? And now we know that these red eyes is appeared by the change of the DNA sequence, right? Because then this DNA sequence change can lead to having the proteins to have red eye phenotype, right? But anyhow, at the time, Morgan was totally shocked and was totally changed, was totally transformed his idea. The reason is that, as I mentioned, that Morgan was a long supporter of the gradualism, right? Evolution has to happen gradually over the generation. But then Morgan saw great exception, which is the red-eye flies just suddenly happening, right? Not reddish, not whitish, between red and whitish. It's just the red eyes, right? So Morgan describing the situation like due to evidently for the sporting. Sporting is interesting word. Sporting is a word totally opposite to the gradualism. This is the situation, the organism suddenly have certain character due to the mutation, right? So this is like, when you see X-Men movie, right? So like, Hugo de Vries or some people, they always thought about this sporting situation, mutation situation, where they describe the sporting as a sortation. Sortation is S-A-L-T-A-T-I-O-N. And this is the opposite view to the gradualism. And Morgan suddenly transformed his idea into the Mendelian field. This is like situation, you believe certain religion, then one day you met something, then you change your total background, right? So Morgan's experiment has that idea, right? So Morgan trained from the gradualism situation, but from his experiment, he see totally different things, right? And then he starts studying the mutation and Mendelian perspective, right? Okay, so Morgan's experiment, he crossed the first generation, which is the heterozygous female, and that is the red eye, one heterozygous, the dominant allele, and crossed with the red eye males, right? So which is the W plus, right? So the first generation, all of your offspring has the red eyes because you have all of this constitute, right? In F2, you have the 3-to-1 ratio of red eye to white eyes, but only observed, only male has the white eyes, right? The reason is that following this situation and the alleles located on the X chromosomes determining that the eye color, and this eye color gene, we call this sex-linked gene, and because of location on the sex chromosome, the ratio having you on an F1 generation and F2 generation is determined by the fact of the chromosome location, right? So the Morgan can conclude that the eye color determining genes has to be sitting on the X chromosome, and Y chromosome does not carry a gene for eye color, right? So this is the sort of the change the notion of Mendelian factors from chromosome to the concept of the gene, right? Because the Mendelian factors is working as a gene, which is determining the phenotype, and that is working in an inheritance situation, right? And he published the paper in Science 1911, and he explicitly state that chromosome carry heritable factors or gene, right? That is great statement at the time. The reason is that before Morgan's experiment, we only knew that the chromosome was the heritable factor, right? But Morgan added one more layer, which is gene, into this situation, right? So chromosome is the place carrying gene, and this gene is the heritable factors that Mendel mentioned, right? also, one more thing discovered by the Morgan was the linked genes, right? So genes are close together on a chromosome. They are tend to be inherited together, right? Now we know that the genetic linkage, right? So linked genes tends to separate from one another during crossover in phase 1. We discussed in a previous slide. And during crossover in prophase, they tend to stay together instead of segregating and switching. And this is the opposition to independent assortment assumed by Mendel, right? And crossover events occasionally separate and exchange linked genes and produce new combinations of genes. And we also learned about this situation, and this is a source of the variation in each generation, right? Okay, so discovery of linked genes. Mendel used the crossbreed of the two traits, which is the D hybrid. And one situation is this, and the other, the organisms is this. And then in F1, the generation expected genotype based on Mendelian genetics was G, two heterozygous combinations of two heterozygous, but he didn't expect, he expected this, but he didn't observe this situation. So if you look at his experiments, he crossed over these two, one heterozygous parents and the recessive parents. But then after a generation, he actually see the 3-to-1 ratio in the generation in the two traits, as if these two traits are the same one. The reason is that the locus determining these two phenotypes, these two traits, are on the same chromosome and they are linked. So there are more chance to have the segregate together during each generation, right? So there was the observation why they have the same the more chance of the segregation in each inheritance, right? So we discussed this issue in a genetic linkage during meiosis, and we talked about the crossover. And after that observation, people start developing how we measure this genetic linkage on the chromosomes, right? Because the physical distance of the two loci has the great chance of having the segregation, co-segregation, right? So using that idea and the measurement, people using the unit called centimorgan, and this is followed by Morgan's name and the frequency of the crossing over, and that is widely used in, you know, whether these two loci or traits are linked or not. And I mean, in these days, we still use this idea for many ranges of studies, right? And the linkage map, also we call the genetic map, is the frequency of this crossing over or recombination of the genes, and we use this information into the measuring the phenotypes or genotypes in each species, right? Okay, so it's a quick recap of today's presentation. We learned about the different terminologies on the chromosomes and genes, and also how Morgan's experiment is linked to the Mendelian factors, and also we discuss about the genetic linkage, the idea development from Morgan's experiment, right? I think this is sort of the connecting dots between chromosome theory and also the mutation topics in session 3, but also you can source that idea from the Mendelian factors and how this is rediscussed over the time, and how early geneticists they see the heritable factors in their experiments. So please keep in mind that the Morgan's experiment and also the terminology used in the time. And lastly, I want to introduce this the review, and this is by Carl Benson in 2001, and it's published in I think it's Nature Genetics or somewhere. Okay, the first screen. I actually want to search by Google. Okay, so this is probably the one. Okay, so this is published in Nature Review Genetics. And this is one of the greatest review I ever found in my life, right? So I think this is, recapitulating all of the important topics in early genetics from Mendel's, Darwin, and also Morgan's idea, and how Morgan see inheritance as a blending, entity at the early time, and how the major mainstream society perceived Mendel's idea and Darwin's idea, and how this idea has been evolved by the experimentalist in early 20s or mid 20s. So please read this article for your understanding, because we learned about the Morgan or whatever the genetic linkage as a high school topics or whatever the exam topics, do very boring calculation. But I don't think this is quite important in modern biology, because no one wants to do the calculation. All computation stops doing that, but we have to know about the implication of how people perceive that Mendel's idea on early days, and what that means in large scale genomic area too. So always important to think about the how early terminology has been formed at the time, and how that is the perceived in these days, still same or slightly different, or we see the new dimension or new places, and thinking about that notion going to give you the next agenda to the next generation as well. So we should discuss the good things from the oldies, and thought has to think about the where we have to move forward, and which topics has to be the studied further in the later time in our days, right? So please sort of clear up your idea about the session 2 using this article, and if you have more questions or comments or unclear stuffs, always leave yours onto the Slack, and let's do the good discussion on there. Okay, so I think that's all. And also, if you have some further questions, see you on Slack, and we're going to continue the stuffs on heritability this Wednesday, and that would be probably the hardest one in this session. So please review the slides before the class, and hopefully you enjoy the lecture in the next time. Okay, so that's everything, and hope you have a wonderful week start, and see you next time.